Um, so thanks everyone out in the internet ether for joining us. I'm Jordan. I'm a professor here at USC, as most of you know, and associate director at CAIS with Eric, Bistra, and Phoebe. Today, you all have the pleasure of listening to one of my really good friends, David Eddy, talk about his research and some really interesting stuff that he's doing currently. I have a whole list of things that he's done. I'm not going to read this list. I thought I was going to. I'll just let you know who he is and tell you some of the cool stuff we've done. David is a research scientist at Mass Gen Hospital in Boston. He works at the Center for Addiction Medicine and Recovery Institute. He's also a licensed clinical psychologist, so he's one of these rare breeds of high-end researchers and a clinician. So he's still in the clinical world, which is great. So he's a clinical psychologist at Mass Gen Hospital in the Department of Psychiatry, and an assistant professor at Harvard Medical School. So he's all over the place. I've known David, gosh, I don't know, man, we're probably going on almost 9 years at this point. Written boatloads of papers together, and recently David and I have struck up an interest with Bistra, who's joining us here, and we're doing a lot of really interesting machine learning stuff. So I won't talk a lot about that, but I believe a lot of what David is going to present today has a real potential to be integrated with machine learning and AI, a lot of the stuff that CAIS does here. So I'll let you go ahead and get started, David. I think we do save questions for the end, just FYI. So there's a Q&A thing at the end. And I think Haley might help organize that.
Thanks, Jordan. That's a lovely introduction. So it's a pleasure to be here. And I just thought I'd start by giving you a little roadmap of where we're headed today. So to begin, I wanted to start by giving everybody a quick overview of the central autonomic network and heart rate variability, because this is really instrumental to the work I'm going to be presenting today. So this is going to be a little bit of a neurobiology 101 kind of intro, but it's important context because otherwise, a lot of my dependent variables are measures of heart rate variability. So if you don't have the broader context, it's going to be hard to follow. So I'll do a little bit of education around central autonomic network and HRV. Then I'm going to introduce a study that we completed a few years ago, but really just getting into the data analysis now and starting to publish some papers. And this is my NIAAA F32 project. So that's the National Institute of Alcohol Abuse and Alcoholism. It was F32 project that sponsored my postdoctoral fellowship at Massachusetts General Hospital and Harvard Medical School. So I'm going to give you an overview of that study. And then I'm going to show you a couple of analyses that we're doing based on that research. One where we're using these neurocardiac or heart rate variability measures to predict subsequent alcohol use amongst individuals in the first year of a current alcohol use disorder recovery attempt. And then, and this is sort of more where the AI machine learning piece, I'm going to be talking about some preliminary analyses we've been doing on this F32 data, identifying stress states using biosensors embedded in smartwatches. And that's actually part of the specific aims of my K23, my NIAAA K23 that's currently underway. But we're really just in data collection for that project. But we got a bit ahead of ourselves, and because we had some useful data from the F32, we wanted to start, taking a sneak peek and seeing if we could use some of the machine learning approaches we want to use in that project on this existing data. Okay, so a quick overview of the central autonomic network and autonomic nervous system. I'm a clinical psychologist, as Jordan said, but I'm also a researcher. I'm very interested in how difficulty regulating emotions can get people into trouble with alcohol and other drugs. I really think of all substance use behaviors as being emotion driven, whether we're trying to boost positive affect through substance use or just trying to mitigate negative affect. Emotions are usually involved in some way, shape or form when we're talking about substance use. And we know that once people have developed a substance use disorder, they become more and more reliant on substances to cope emotionally. Now, a lot of previous research on emotion regulation more broadly, and emotion regulation in the context of substance use disorders, has studied emotions at the level of the brain and of the mind, which is super, super important. And in fact, the central autonomic network is a constellation of brain structures that's been identified that really is responsible for how we process emotions, how we experience emotions, how we regulate emotions, but also how we regulate behaviors. Now, the central autonomic network includes a constellation of brain structures, and this is really an oversimplified cartoon. It's a little more complicated than this, but it's important to know that the central autonomic network connects with the autonomic nervous system, which I'm sure everybody's heard of, to communicate with the body. So the central autonomic network, it's not just helping us regulate our emotions and our behaviors at the level of the brain. It's affecting dynamic changes in our physiology, moment to moment, changes in hormone levels, for instance, through the endocrine system, and changes in heart rate and blood pressure, heart rate variability, which I'm going to be talking about. So although the autonomic nervous system, I feel, really gets a lot of the attention, that's what we talk about a lot in psychiatry and psychology, actually, the central autonomic network's doing most of the work. The autonomic nervous system is really just the electrical pathways that allow the brain to communicate with the body. So I just dropped this slide in here because it's relevant. I think, so I'm going to be talking about addiction today, and this is from a review paper I recently published with my colleagues Marsha Bates and Jennifer Buckman at Rutgers in addiction biology. And what we did is we took the Kubin-Volkow sort of well-articulated neurobiological model of addiction, which is based on the work of like thousands of people, but they've really done a nice job of synthesizing a really large body of research trying to understand the neurobiology of addiction. And what we have here is sort of the primary areas in the Kubin-Volkow model in blue, green, and red. And over that, we've overlaid the areas associated with the central autonomic network. And what you see is there's just a tremendous amount of overlap. So to me, this really speaks to how important not only neurological brain level processes are in addiction, but also the brain-body communication, and body-brain communication. We think it's bidirectional communication pathways get crossed up in addiction. So we don't just have problems at the level of the brain, we have problems at the level of the body as well in addiction. And the more dysregulated these systems get and the worse their communication, the more and more difficult it gets people to regulate their emotions. So now we're sort of segueing, the heart rate variability here. So I told you the central autonomic network is connected to the cardiovascular system through the autonomic nervous system. So nerve fibers descend through the parasympathetic, like the rest digest branch, and the sympathetic pathways, the fight or flight branch, to synapse on the atrioventricular and sinoatrial nodes of the heart. And you can almost think of these as sort of clusters of neurons that are summation points in the heart, which are receiving information from the central autonomic network through both the parasympathetic and sympathetic branches to make decisions on speeding up or slowing down heart rate, but also to affect blood pressure through arterial dilation or constriction. And so you've got the heart is being duly innervated by the central autonomic network. And what that means is we can calculate measures of heart rate variability, which actually can tell us a lot about what's going on in the central autonomic network. The value of this is that we can study what's going on in the central autonomic network at relatively low cost, non-invasively, and also we can study what's going on in the central autonomic network in real time. Because we can measure ECG in the lab, we can also measure ECG in real time using miniaturized devices, which can monitor people's heart rhythm throughout the day. So heart rate variability, or really just a variance in heart rate, is a powerful tool for understanding what's going on at the level of the central autonomic network. So to measure heart rate variability in its most simple form, you could say, take the standard deviation. So here we have a diagram of an ECG waveform. Most of you are probably familiar with this waveform. You know that these spikes here represent a systolic contraction of the heart. It's called the R spike. And here we have labeled 845, 745, 812. That's just milliseconds. It's just measuring the distance from R spike to R spike. And you could calculate mean heart rate from this information. You could also calculate the standard deviation of heart rate. But in fact, you can calculate a lot of other measures. These are just some common ones. It's probably about 100 HRV statistics that we commonly use. These are some of the big ones you see over and over again in literature. RMSSD and PNN50 are really measures of parasympathetic activity. So depending on how we slice variance data, we can determine, we can make inferences about what's going on in both the parasympathetic and to a lesser extent, sympathetic branch of the autonomic nervous system. So that's in the time domain, and I won't get into this too much, but in the frequency domain, we can do fast Fourier transforms of the variance data and create a spectral representation of the variance. It's essentially histograms, and we can look at the area under the curve and calculate the area under the high frequency curve and low frequency respectively. And also these can also tell us for high frequency heart rate variability about what's going on in the parasympathetic branch of the autonomic nervous system, and low frequency really captures both parasympathetic and sympathetic activity. Now, heart rate variability is a widely accepted, widely used biomarker of health, right? So we know that people with higher variance in heart rate are typically healthier. What we consistently see is that people with psychopathology like mood disorders, substance use disorder, PTSD, consistently have lower variability than healthy controls. Similarly, people with physiological conditions, medical conditions, will often have lower heart rate variability. And this maybe goes against what I think most of us would intuit. I always thought a slow and steady heart rate would be a healthy heart rate, but in fact, variance is good. Variance is indicative of the brain and the body responding dynamically to situational demands, whether that's a stressor or if it's just having a conversation with a friend. We're constantly modulating heart rate and blood pressure to meet situational demand. And so for the ability of the brain and body to modulate heart rate in a dynamic way, and as well return to resting baseline quickly after it's been perturbed, maybe by an acute stressor, that's really important as well. What we see in people with psychopathology is they have lower resting heart rate variability, typically higher heart rate, and as well, in a lot of conditions, once perturbed or once stressed, it takes folks longer to get back to their resting heart rate, right? So we think of this as a loss of biobehavioral flexibility. Okay. So let me jump into the F32 study I briefly mentioned in my introduction. So this work was funded by NIAAA. I was also funded by competitive awards from Harvard Medical School and Harvard University. And in this study, we recruited to 42 treatment-seeking adults between the ages of 18 and 65 with alcohol use disorder who were in the first year of a current alcohol use disorder recovery attempt. Now, folks had to have a goal of alcohol abstinence to be included in the study. We allowed most medications because we wanted a fairly naturalistic sample, but we did exclude folks taking medications directly affecting the heart, like beta blockers, which are a class of medications that people commonly take to manage hypertension or high blood pressure. But psych meds were okay for the study, and psychiatric comorbidity was also allowed. So in our study paradigm, participants came into the lab on study day one, did some baseline questionnaire measures, and we installed an ecological momentary assessment app on their smartphones. So this app essentially pinged people 3 times a day at random times to ask them to do little mini 1-minute surveys on their emotional state in that moment. And I'll show you what that looks like on the next slide. So we did that for 6 days, but we also had people wear an ambulatory ECG monitor, which again, I'll show you on a subsequent slide, small lightweight device that folks wore just for the first 4 days. We considered having them wear it for the full 6 days, but it's a lot of time to be wearing an ECG device, even though it's fairly lightweight and fairly comfortable. We didn't want folks to burn out and drop out of the study. So we limited ourselves to 4 days of ECG monitoring. That still gave us plenty of data. So then after 6 days, participants have brought back the device. We don't see them for 90 days. And then at 90-day follow-up, we do the same assessment, questionnaire assessment battery we did at baseline. And at 90-day follow-up, we also do 90-day timeline follow back of alcohol use to assess if and any alcohol use for the past 90 days. Okay, so let me just show the EMA app, just because I think visuals can be helpful. This is the screenshots of the app here. Folks rated their craving, stress. We asked them about their confidence to remain abstinent. They just had a Likert scale, and they just slide the point on the bar there, and they completed 3 of these a day. We also assessed positive affect as well. We asked about happiness. ECG device we used was a Bitium Pharos 180, which is a small lightweight device that can be worn on a chest strap. You can also use fast fix patches, like giant band-aids that are going to fix these devices as well to the chest. We were using the chest strap in the study. But actually, these devices are pretty good. They have a long battery life. They can continuously run for almost a week. They have huge memory capacity so that they can continuously record ECG data. And these are huge files, by the way, because we're sampling at, maybe 500 hertz. So, that's 500 samples a second. You can imagine over the course of several days of recording, you've got a really large file. But even with some participant movement, we often get pretty good clear ECG waveform. You see that on the top line here. Hopefully you can see my pointer. And here as well, you'll see 3-dimensional accelerometry on the X, Y, and Z axis. So this device not only does it monitor heart rhythm, ECG, electrocardiogram, it also measures movement. And that's important in these kinds of ambulatory HRV studies, because obviously movement affects heart rate and heart rate variability. So we want to be able to monitor movement so that we can statistically control for that later on. Okay. So let me just describe our sample. On average, about 41, 42 years old. Our sample was primarily male with 38% female. Racially, we had around 19% Black or African American identifying people, about 74% identifying as white. We really, this is a pretty typical racial spread of a study conducted in Boston, but I think we still have a ways to go to try and recruit more diverse samples. So let's look at baseline alcohol use in the past 30 days. I didn't mention this, but we did a timeline followed back at baseline. And on average, folks reported around 88% days abstinent, those days not drinking in the past 30 days at baseline. And then over the 90-day follow-up, after they participated in the ECG and EMA paradigm, we saw again around like high 80s at 90% days abstinent, with 55% of the sample remaining totally abstinent through that 90-day follow-up period. Okay. So the first question we asked, we had a bunch of specific aims in this study, but this is one of the most important questions we wanted to ask. Can you use heart rate variability to predict subsequent alcohol use? Like why? And so why do we care about this? Well, we have a lot of self-report measures that are predictive of subsequent alcohol use amongst treatment seekers, but we don't really have many, maybe any, objective biomarkers of risk. So this could be something, not to replace subjective measures, self-report measures that could be predictive of relapse risk, but to really complement self-report measures. Because as we know, a lot of folks in early recovery from addiction don't have great self-awareness, interoceptive awareness of their own affective states and their own risk. So if we had an objective biomarker that we could fairly easily assess at relatively low cost, and we can, because these devices have miniaturized, they've gotten very affordable, and we can assess heart rate variability in the clinic now at really low cost very quickly. So I wondered, could heart rate variability, being an indicant of biobehavioral flexibility, predict who remains abstinent and who lapses to alcohol use? So just to quickly show you our models here, we were using heart rate variability to predict follow-up % days abstinent in our first set of models. And then in a subsequent set of models, I wanted to see if baseline drinking might moderate the association between heart rate variability and follow-up % days abstinent. So first, is there a raw association between heart rate variability and subsequent alcohol use? But then, does baseline alcohol use moderate that relationship? And here's what we found for the bivariate associations. So we found significant positive associations between our parasympathetic indices, or primarily parasympathetic indices, and percent days abstinence. So what that meant, and this is as we hypothesize, folks with higher parasympathetic HIV, i.e. more biobehavioral flexibility, were more likely to stay abstinent. They drank less over follow-up. Conversely, folks who had higher sympathetic activation, which we often associate with chronic stress and less biobehavioral flexibility, those folks had lower percent days abstinent over follow-up, i.e. they drank more. So then the subsequent question was, well, does it matter then how much folks were drinking at baseline? Because that's always an important consideration. Obviously, baseline alcohol use and follow-up alcohol use or subsequent alcohol use is almost always positively correlated in substance use disorder recovering populations. And that's what we saw here, as we would expect. So we saw a positive association between baseline percent days abstinent and follow-up percent days abstinence. But in fact, baseline percent days abstinent also moderated this association. And so what that meant is that folks with lower baseline percent days abstinent, folks who had higher heart rate variability, parasympathetically mediated heart rate variability, that seemed to attenuate or mitigate the positive association between baseline drinking and follow-up drinking. So folks with high heart rate variability were essentially, it seems, able to shift sets, change their behavior, and reduce their drinking. But for folks with low heart rate variability, the relationship was almost strengthened between the likelihood that they were going to drink actually seemed to be more robust. And we saw a similar effect for our sympathetic measures of heart rate variability, except it's flipped, as you would expect. So we saw exactly the opposite. So folks with higher sympathetic arousal here in the purple dotted line were more likely to continue drinking, whereas folks with lower sympathetic arousal tended to do better over follow-up versus baseline. So we wanted to do some post-hoc checks to see if potentially other factors might have been influencing these results. These were fairly consistent and robust results. So first up, we looked if drinking goal over follow-up mattered. So as mentioned at the beginning, everybody had to have an abstinence drinking goal to be in the study because we wanted homogeneity, right? So that if people were drinking, we didn't want it just to be because they were moderating or wanted to moderate. Unfortunately, 5 people in the sample changed their mind. During the 90-day follow-up period, as tends to happen, they went from having an abstinence goal at the beginning of the study to having a moderation goal. And so what we did, we entered that in the model as sort of a binary, did they change to an abstinence, to a moderation goal or not? And that didn't affect the results. We also controlled for movement. Like we thought, well, perhaps, perhaps movement's affecting these associations in some way. Maybe that's accounting for the correlations we're seeing, but no, movement didn't seem to matter. Respiration, even though we didn't directly measure it, you can infer it from the ECG waveform, didn't seem to be affecting our results. And we also controlled for depression using measures of from the Beck Depression Inventory, didn't see any effect there either. So in conclusion, these findings suggest that show preliminary evidence that heart rate variability has some utility as an indicator of alcohol use risk in folks in early recovery from alcohol use disorder. And I think these findings also underscore the potential utility of interventions that are body-based, like heart rate variability, biofeedback, and resonance frequency breathing, which I haven't talked about at all today. Some of you might be familiar with these interventions. For those of you who aren't, these are breathing-based interventions that take advantage of the relationship between the respiration and the cardiovascular system to affect changes in heart rate variability, acute changes in heart rate variability, which seem to have salutary effects on the mind and brain and help people buffer stress more effectively and regulate their emotions better. And we know there's a 5, 6, 7, 8 maybe now studies applying heart rate variability biofeedback as a clinical intervention for substance use disorder, and all have shown really promising results in terms of helping people reduce craving and managing their stress more effectively. Okay, so let me jump in now. I think we're going to save questions to the end, right? So let me jump in to another set of analyses we're doing, again, using the same data set I've been talking about here. The goals of these analyses are really from my IEEE K23 study, where we're trying to identify stress states using commercial biosensors. And you might say, well, like that's a weird thing for NIAAA to be funding. They're interested in addiction and addiction recovery. What does this have to do with either of those things? Well, let me tell you. First up, though, let me just mention these analyses were done in conjunction with Dr. Richard Fletcher from the Massachusetts Institute of Technology, and a really talented undergraduate working with Dr. Fletcher, Ming-Yin Yang. So I should just, with a caveat, the analyses I'm going to be showing you were largely done by Richard and Ming, and so I'll do my best to answer questions, but I'm sure a lot of the people here on this talk are more expert in these analytical methods than I am. Okay, so why real-time stress detection? Well, as I mentioned earlier, folks in early addiction recovery often have poor self-awareness, or technically speaking, interoceptive awareness of affective states. Folks have difficulty labeling emotion, and if you can't label or identify emotions, that can get you into trouble, especially in early recovery from addiction, because when people with in early recovery experience, say, high levels of negative affect, that often manifests or leads to cravings or urges to use, because that's historically how folks have managed to cope with negative affect. So what would it, that we think there would be tremendous benefit if we could help catch stress, maybe that's even occurring outside of conscious awareness, to let people know they're experiencing high levels of negative affect and to suggest that they do something about it. So the idea being we could use the biosensors embedded in smartwatches and fitness monitors like Garmin devices, Fitbits, Apple Watches, Samsung Gear smartwatches, to monitor affective arousal in real time. And if acute stress states are detected, obviously in the absence of movement, it could trigger these kinds of relapse prevention apps. And this is the HS app from the University of Wisconsin, which is a really awesome relapse prevention app that has a lot of tools that can help people connect to their recovery supports. It has CBT, cognitive behavioral therapy related tools, mindfulness meditations, all kinds of excellent features that can help people to recover. Problem is, though, with these apps, people often forget to use them, right? That's the challenge. And I would say, as well as a clinician, one of the challenging things about working with this population is that although folks will often demonstrate mastery of a skill or a strategy, and I primarily work in a cognitive behavioral therapy framework, so I'm all about skills and strategies and tools, although folks often have a good sense of what to do with these tools when they're working with you in session, so they understand the skills at a cerebral level, often they have trouble applying them in real time when effective levels are high, where the rubber really meets the road. So if we could catch people's stress states, perhaps before they're even aware they're stressed, and tip them off using apps like HS or really any relapse prevention app, that could potentially buffer folks against lapses to substance use. Now, I'm not pretending you're gonna, every time somebody experiences stress that they relapse or lapse to substance use. That's not at all the case. But if we can bring people's awareness to their affective state, we can increase self-awareness, which can, I think, have a salutary effect more broadly. And we might also help prevent some lapses. And as you probably know, in the context of substance use disorder treatment, preventing one relapse can have a huge impact. Or one lapse, I should say, because one lapse can easily lead to full-blown addiction relapse. So we think about lapses as a return to, as a substance use episode, a relapse to a return to active symptomology of substance use disorder. So I think there's real value here, and particularly because we can do these things at relatively low cost. People already have smartphones. A lot of people already have fitness trackers and smartwatches. So if we could create an app to go on people's smartwatches that could be free from the from the Apple Store or the Google Play Store, or maybe just $1 or $2, and they can get these recovery apps at relatively low cost as well, then we could potentially boost people's recovery chances at a fairly low cost in a scalable way. Okay. So we're not the first people to try and predict stress using physiological data. A lot of work, around 100 published papers to date, have utilized primarily heart rate to try and detect stress in real time. Heart rate's really been the focus because we can measure that a lot more reliably using wearable biosensors. Heart rate variability is much more susceptible to noise and movement and missing data. And so it's less reliable on wearable devices, but we're working on that problem now. So generally, the results of previous published trials have depended on the training data used in the machine learning models. So these papers, they've all created some kind of algorithm for stress detection based on real-time heart rate. They found the greatest accuracy where the machine learning models have been trained using a single individual. So that's where you take a single individual, measure their heart rate and their response perhaps to a stressor, and then train an algorithm specifically for that individual. Well, it's really accurate, but it's not really scalable. Like, it's very hard to do that at scale from an app that people are going to download from the App Store. So what we can do is we can create a ground truth of stress reactivity using a sample of individuals that can represent the population. And we can use lab-based paradigms where we can expose people to a series of stresses. Usually we measure them at resting baseline, their physiology, and then we expose them to an acute stressor in the lab, and we can measure their stress reactivity. And so machine learning models based on these lab paradigms are pretty good at detecting stress in real time, 80 to 90%. Less reliable are machine learning models that are trained on self-report measures. That's where we take physiological data that's been labeled using self-reported stress using ecological momentary assessment. So around 70, 80% accuracy on average. So we wanted to jump in and see if we could improve stress detection, current stress detection algorithms by focusing not so much on heart rate, although we are including measures of heart rate, but also on heart rate variability. We think heart rate variability is probably a better indicant of stress than heart rate, because to see an acute increase in heart rate, one tends to need a significant stressor, whereas heart rate variability will change or respond to a lower level stressor. And so heart rate variability is perhaps a more sensitive indicant of an acute stress state. So first up, before I get to the data from my F32 study, we wanted to test out some of these models using a lab-based paradigm. So we took some publicly accessible data from the Wearable Stress and Affect Detection Dataset, known as the WESAD. This was a sample. This is a data set created from a fairly narrow age range of individuals, 25 to 31, so young people who went through a lab-based cue reactivity paradigm where they were measured at rest, during a meditation task, during an acute stressor, and also during an amusement task. We really just focus on the at rest and the stress tasks. They used electrocardiogram recordings throughout these 4 tasks. They also asked folks to self-report their emotional states based on using the positive and negative affect schedule, the PANAS, which is a really widely used self-report measure of emotional state. So we borrowed this data, and first of all, we used TSNE approach to distribute on these measures 20th, 80th percentile of the inner beat interval, SDNN, high frequency HRV, the low frequency and high frequency ratio, and mean heart rate. We essentially just told the model to create 2 clusters because we anticipated we're going to have a resting group and a stress group. And each of these dots represents an individual at rest and during a stressor. So we essentially have 2 dots per individual. Right. And we just used different clustering techniques, spectral clustering, Ward clustering, Birch, and Gaussian mixture to split our samples. into two distinct groups. And in theory, because one of the tasks was stress and one of them was relaxation, the split or the clustering should map on to our resting and stress states. So did they? That's the question. They did pretty well. And I just showed here the coefficients as well to show really what was driving the clustering. The 20th percentile of the interbeat interval and mean heart rate and the 80th percentile seem to be really driving the effects. And I should say we just really came to these, use these features from trial and error. We had some a priori ideas of what would work, but we just tried a bunch of different models to see what would give us the best prediction, best clustering. And so on the left here, you have the spectral clustering, and then we've relabeled the dots here in the same color code, which is a bit confusing. But here on the right-hand side, we've relabeled the dots based on the task or the condition. So orange is the stress task, blue is resting. And what you'll see is our spectral clustering did a pretty good job of actually splitting our different conditions here. And in fact, when we use a logistic regression model to see how the area under the curve, we actually see almost 90% the variance accounted for, which is actually pretty good, which is sort of what we typically see in the literature based on Q-reactivity, machine learning algorithms trained with Q-reactivity paradigms from lab-based research. Okay, so the question is, how did the same model hold up using our ambulatory data, which involved measuring ECG in real time as people navigated their world and using the EMA self-reported stress to label our data? Well, first up, we just asked the model to create two clusters, hypothetically stressed and not stressed. And so each of these dots represents a distinct EMA episode. So each participant probably has like 12 to 15 dots represented here. And so we use some of the same measures, again, mean heart rate, 20th and 80th percentile, but also we included age in our cluster analysis because we had a much more heterogeneous age range of participants in our data set compared to the WESAD. And also we discovered that body mass index varied greatly in our sample, whereas in the WESAD it didn't. So we included BMI in our analysis as well. And so heart rate was really, heart rate, 20th and 80th percentile, and age really seemed to be driving most of the effect. So now what happened, if we then relabel our dots with low stress or high stress, and we had to create an arbitrary cut point here. So we said EMA reported stress of less than 2 out of 10, equal to or less than 2 out of 10. We said that's low stress, and high stress is 3 or greater. Maybe not the greatest cut point. That's something we have to figure out and we're playing around with now. But these are just preliminary analyses. And in my K23 project, we both have ambulatory data and lab-based data. So we're going to actually try and train two models and see which one performs better using the respective lab-based or ambulatory data. So what you see here, unlike with the WESAD trained model, we don't see great specificity. You've got a mix of blue dots here and there, and the orange dots are a little all over the place. And in fact, when we look at the area under the curve, just skipping ahead a little bit here, we're down to 0.63, which versus 0.89 for the WESAD ground truth. So that's a significant loss of specificity. So we asked ourselves, why is that the case? Is there just something fundamentally like, is it just that EMA data is like less reliable? And that might be the case. That seems to be what we see in the literature. But we also thought, well, we've got a real range of BMIs in our sample. Maybe BMI is a factor here. So Ming, what she did here is she essentially separated out people who had BMIs indicative of obesity versus folks who had a BMI in the recommended range. And actually we saw really different spectral clustering, which suggests to us that in our future models, controlling for BMI is going to be a really important thing to do. So another challenge with EMA trained models is that self-reported stress is highly idiosyncratic. So this across the bottom axis here, we have participants 1 through 43. We ended up with 42 in the sample. We had to exclude 1. But you see there's a lot of variance here, and these are the interquartile plots. I believe the green triangle is indicative of the mean, and you've got your 25th and 75th quartile ranges, and you've got your outliers, your standard deviation indicated here, and these are your outliers, the dots. So you just see, there's a lot of variance here. And so one of the things that Rich Fletcher is working on now is normalizing self-reported stress data like this to see if that can increase the specificity of our models. Okay, so I think that's it. I'm going to stop there. I just wanted to thank my funders for this research, NIAAA, Harvard Medical School, Harvard University. Also, really wanted to thank my collaborators on this work, Sarah Wyman, Agata Pietzak, and Zadie Zay, who are really instrumental in doing the work and the first analyses that I presented. And of course, I wanted to thank Richard Fletcher again, Ming Yang Ying for their work on the second phase of the analyses. And I'll leave it there and open it up for questions.
Great. Thanks a lot, David. Well, I got quite a few questions, but I'm probably going to hold off to see. I actually can't see any of the Q&As. And I don't think I can even enter them into the thing. So
maybe if anybody has a question, they can just unmute. And
yeah.
well,
Maybe I can also just get it started too, instead of me trying to click around on this computer. So with the in this last study with stress and using ML, I have a really simple question, then one that's maybe more, I don't know, thought-provoking. The simple question is, why did you force 2 clusters? Why didn't you allow the data to simply extrapolate to whatever clusters they wanted?
in the WESAD analyses?
Yeah.
Right. So we expected to see 2 clusters because the models were trained on data representing folks under resting conditions, under stress conditions. So in theory, we should have seen 2 clear clusters. But you make an excellent point. Just because we think it should be so doesn't make it so. And in fact, this is where we have to take this work. And I think this is the important work we need to do now in terms of understanding moderators.
Yeah.
It might depend, right? People at baseline can look different from one another. People under stress conditions can look different from one another, right? So this is a very crude and rudimentary preliminary set of analyses. And we want to get a lot more granular with these. And, these are really, I don't even like really calling this machine learning. I mean, really, this is essentially a cluster analysis. This is sort of the precursor, the preliminary work, I guess we do before we get to developing our machine learning models.
Yeah. And I think my next question is like, okay, so I figured that was the case. That's all. That is a testable theory, though. We expect 2. What do we really get? Which is an interesting question in and of itself. But like, so let's just say there's 2 final clusters. I'm almost wondering if it's possible to use some of the methods that me, you, and Beaster have been using, and John Prindle, using SCM forests and sort of these penalized regressions to basically say, here's our clusters of people, A and B. Now, what are our best predictors of that? What are our top predictors of those? So now the cluster becomes the outcome, right? And it's, I know people get upset about that, the classify, analyze thing, whatever, just do it. But like, now this is your outcome. Now we can say, here's our top predictors of these things. And then here's the valence of that effect with that that lasso regression.
I like it. And we've really forced a binary outcome in our models of the WESAD data there, because we're expecting a binary outcome. But I think in reality, you know, a real-time stress detection app, I think is not going to be, I don't imagine that it's going to produce a binary outcome of stress or no stress. I think it's actually, I'm imagining we'll probably have grades of stress, so that we'll have probable low stress, probable moderate stress, and probable high stress. And then the decision will be, at what point should we trigger a just-in-time intervention? At what point do we have the app intervene? Do we wait until the stress is high? Maybe we let the wearer, the end user, decide at the threshold at which they want to be notified based on their own sort of self-awareness of you know, how good they are with this stuff.
Yeah. Eric has a question, I believe.
Eric,
Hi. I mean, it's also maybe partly comment, partly just, you know, musings, but I found this to be an absolutely fascinating talk, and I was really struck by when you were showing with the first study, the really strength that you had in predicting sort of percent days used as a function of the heart rate variability, and then kind of the challenges that you're facing with, you know, using the commercially available monitors to really get beyond, you know, heart rate into heart rate variability. And I think one of the things that you said, which is what prompted me to raise my hand when I did, was something about lower stress events versus acute stress events, maybe chronic stress events, and kind of all of that wrapped up with the notion that you were sharing about, you know, people in early recovery not being very good at identifying their emotional states, not being very good at necessarily assessing sort of their stress levels. And I'm wondering if part of that is like somehow wrapped up in, it might be not so difficult for people to identify, hey, I'm really stressed out right now. And it's more challenging for people who are in recovery to identify these sort of moderate stress events that might be more picked up by the heart rate variability. And part of your inaccuracy that you're encountering here is just like, you're kind of measuring the wrong biophysiological phenomenon in some ways with the current technology. And I'm just kind of wondering what your thoughts about that moving forward are. And I may have missed a key variable in the whole discussion there, so forgive me if I've misrepresented what you said.
No, you highlight a really important consideration. And this is one of the challenges. I mean, we're studying very meta concepts here, right? Stress. What is stress? And we know stress, whatever it is, you know, how we operationally define it, can be experienced acutely and chronically. And that can influence behavior in different ways. You know, how people respond to stress also varies greatly. One thing for me clinically that I consistently observe is that, you know, yes, people have poor interoceptive awareness of affective states. That doesn't mean that people with, you know, an early recovery aren't experiencing high levels of negative affect. I think often it comes out in other ways. So for instance, you know, I might have a patient who will say, you know, doc, I was doing really well. And then just out of nowhere, you know, I just started experiencing these cravings, and I was really overwhelmed. And usually when we drill down into it, the cravings didn't come out of nowhere. Often they're a response to either an interoceptive or exteroceptive environmental stress or cue stimuli. Right. And so, you know, sometimes we're identifying high levels of negative affect, sometimes, you know, stress or anxiety that's really probably driving the craving. But what folks experience is the craving and the discomfort associated with that, but they don't observe the intermediary step that led them to the craving. And I think that's where the risk is, because that's really the point we want to be intervening. It's like we want, if we're experiencing elevated stress, we want to catch that early and often and be proactive about it so that we can minimize craving or at least manage it more effectively. But this is an incredibly difficult problem. And as well, heart rate variability, it's a biomarker for overall health. And one of the criticisms is like, well, it seems like heart rate variability does everything, right? It's an indicator of health. It's this, it's that. It's like, yeah, okay. You know, we're not saying it's exclusively a biomarker of drinking risk. We're just saying it's a biomarker of biobehavioral flexibility that we can leverage to suggest potential future behaviors. Yeah.
I think, oh, go ahead, Eric. Yeah.
But no, I was just going to say, like, there was another angle to this that I thought was really interesting that I just wanted to throw out there, which was the intervention implications that you were discussing at different points, right? So sort of the with respect to the biofeedback and people learning to sort of have more heart rate variability, I suppose control. And then also when you were mentioning, with the just-in-time apps, some of the features of that being things like mindfulness, where, I know that sort of, I mean, Jordan's the expert on mindfulness. But I know that this is, part of it is also sort of, affective, control, which also has a lot to do with, breathing and which then, as you were pointing out, has heart rate control. And so, I mean, it's interesting to think about these interventions that have not maybe had as clear of sort of biological etiologies. You're also starting to uncover why it is that they might be helping with some of these, especially in the context of addiction and recovery. So I thought that was really cool, and kind of curious about, like, you as a clinician, like, what's your desire about where do you want to go intervention wise with this work?
Yeah, thanks. I mean, there's a lot of really excellent work done at the level of the brain and mind, the cognitive behavioral therapy, dialectical behavior therapy, and I even think would classify mindfulness and in that, put mindfulness in that basket as well, like increasing awareness of what's arising in the moment so that we can act more effectively, be less reactive and more responsive to what's going on. So I think a lot of excellent work's being done in those domains, and a lot of my work as a clinician is based, on cognitive behavioral theories and intervention strategies. But I think what's being left out, because we have tended to hyperemphasize or focus mostly on the brain and on the mind, we're leaving out potential opportunities to intervene on problems like substance use disorder at the level of the body, because we know physiological dysregulation is a huge part of it. So what if we can have people do a breathing exercise that we know helps re-regulate the central autonomic network and autonomic nervous system by the body-brain communication pathways, specifically the baroreflex communication pathways from the body to the brain. Can that add something in addition to our cognitive and behavioral strategies that we're teaching folks, give them another tool that they can use in the moment? And the beautiful thing about heart rate variability biofeedback is that it seems to help people when they practice it chronically. So if you do just 10 minutes of practice a day, really not too much, you can see significant reductions in negative affect, greater ability to buffer stress, and pretty good effect sizes as well from very little work. So I think there's tremendous benefit there, and it's something different we can offer people, and it's something tangible and concrete as well. It's a simple breathing practice, like anybody can do it. You don't need. There's not much to wrap your head around, and you'll probably get the benefit. And importantly as well, it can be used acutely when we're managing acute stresses, acute anxiety, fear. And we can use these interventions. And in fact, I've had in a couple of clinical trials of HRV biofeedback we've had anecdotal reports from participants that they're using this strategically to manage panic attacks. They use the breathing practice acutely in the moment when they notice the early warning signs of a panic attack. And I remember one participant distinctly said to me, he said, I could feel it coming. I know the early warning signs of a panic attack, and I could feel one coming, and I did 5 minutes of the breathing, and I didn't have a panic attack. And then he said subsequently he did it again. The next time he noticed a panic attack coming on, and as a result, he felt more agency or control over his panic symptoms.
That's great. This is a perfect segue. We have two questions. I'm going to combine them here for you, sure. David. I'll read a little bit off, but I'm just going to combine them. And one of them is actually about symptom or understanding triggers, and paraphrasing, it's basically saying, do you see the future of this technology is not just being able to predict or prevent, but as a tool for people to be more introspective about their own triggers? And then secondly, following up, so triggers, is that what it's used for? And then secondly, this in this sort of EMA, were you able to collect data about social context? Because that seems really important in terms of, like, who they're with, what they're doing, social behavioral interactions, interventions, et cetera.
Oh yeah, great questions. To the first one about can these kinds of just-in-time interventions not just maybe help get out in front of a potential lapse, but also increase self-awareness of affective states? I think absolutely yes. I mean, the data still need to be collected. We haven't had there aren't a lot of studies testing these just-in-time interventions, but I think inevitably, if we're bringing people's attention to their stress, we're training them inadvertently to become more aware of their stress. Now, that might actually increase their subjective stress levels acutely, but in the long run, that's probably going to help people because eventually they're not going to need the app to notice when they're stressed. And, mindfulness can be brought to bear here as well. And that's another tool we use to help increase people's self-awareness of their states in the moment. So again, mindfulness itself is not an intervention. It's a tool that can help us be more aware of the facts in the moment of our experience so that we can be more effective and respond more effectively to whatever is arising in the moment, whether that's something going on in our environment that's giving us grief, or even an emotional state that's problematic for us. And the second piece about situational factors, excellent question. There were some things, like looking back on that study on the F-32, that I wish had have asked about, but didn't. But one thing we did remember to think about asking about was situational factors. And I didn't show this when I presented the EMA battery, but one of the questions we asked was, where are you in this moment? Just to get a bit of context. Now, it had to be fairly crude. We didn't want to have to people to have to type in something. So we gave them a range of about a dozen options: at home, at a social event, out in public, i.e. on public transport, at a doctor's office, in a meeting, like a mutual help meeting. So we gave people a bunch of options. So we have that data. We have not jumped into that yet, but I think I think there's going to be a lot of value in that. Now, will that have utility in the context of a real world just-in-time intervention? Like, probably not in terms of having people self-report where they are every half hour or something. Like, nobody's going to want to do that. But we might have GPS data that could tip us off to situational factors. We could use geofencing, geolocating, to indicate if people are in high-risk locations. So I think there's a lot of potential there. And that, again, gets to sort of how do we dial in these algorithms and address moderators?
Yeah. Well, I think that brings us right to the hour mark. So we'll just go ahead and end there. I don't see any other questions. So David, thank you so much. As always, you're doing stuff that I can barely wrap my head around, but very important stuff. So I'm excited to see how this works out. And thanks everyone for showing up. David, we'll see you in a couple of days. And
thanks everybody. It's a pleasure.
Okay.
Thank you so much, David. Wonderful
Bye.
talk.
Yeah, thank you so much. It was inspiring.
Thanks, guys.